Hey everyone, welcome back for part 2 of Muscles 2. Yesterday in part 1, I originally said we'd take care of this lecture in 2 different segments. I changed my mind. I think we'll do it in 3. So for part 2 here, we're going to discuss strictly just the neuromuscular junction, pages 82 through 84 in your lecture notes. To pick up where we left off from yesterday, let's remind ourselves of the action potential. The action potential is the electrical signal that nerve cells use to communicate with other cells. It'll also be found again in the skeletal muscle fiber that will produce muscle contraction. Reviewing the steps from part 1, we realized that the cell at rest has a negative membrane potential. That resting membrane potential, as you can see, is about negative 70 millivolts. It's the result of different ion concentrations across that membrane, more specifically sodium and potassium. When the nerve cell or the muscle fiber receives a signal, a neurotransmitter will bind onto its receptor site. That receptor site is actually a ligand-gated sodium channel, and because sodium has a high concentration outside both of those cells, low concentration inside, sodium will start to leak in. As sodium starts to leak in, we're introducing positive charge to inside the cell, and so the membrane potential begins to migrate to a more positive value in a stepwise fashion. We call that a local depolarization, as you can see right here. If we get enough local depolarizations, then we're able to achieve threshold. Threshold we define is at negative 65 millivolts. Once we hit threshold, that is the signal to open up the voltage-gated sodium channels. Because sodium again has a high concentration outside, a low concentration inside, we flood the cell with sodium. As we flood the cell with sodium, we flood the cell with a positive charge. The membrane potential skyrockets to a plus 30. We call that a rapid depolarization. That is actually your action potential. We started at a negative 70, we propagate it all the way to a plus 30. That is a reversal of our membrane potential. That is your action potential. At plus 30, however, some other important events occur. That is the signal, though, for the voltage-gated sodium channels to close, the voltage-gated potassium channels to open. Potassium having a high concentration inside, low concentration outside. Potassium will rush out of the cell. We call that a huge efflux of potassium, carrying positive charge with it, and the membrane potential plummets back down to a negative 70 millivolts. At negative 70 millivolts, that's the signal for the voltage-gated potassium channels to close, and the sodium-potassium pumps reestablish their gradients. Meanwhile, what you've done is you've initiated that action potential right here at the top of the axon in the motor neuron. That action potential will propagate down the axon of the motor neuron until it arrives at the presynaptic or axon terminals. So what we need to figure out is how can we get that action potential from the motor neuron to the skeletal muscle fibers. That is the next step, and that'll be the focus here of the second video on our Muscle 2 lecture. What we do is we use a special term called a synapse to describe the connection between a neuron and the next cell in line. That next cell in line could be either a muscle fiber as you see here, it could be a gland, or it could be another neuron. It all works the same. As we look at a synapse is made up of 3 major parts. The first part is the presynaptic or axon terminal. That's part of the neuron, of course. The second part is a microscopic space that we call the synaptic cleft. It is microscopic. It's thinner than a sheet of paper, but there is actually a physical space between a neuron and the next cell in line. The third component is what we call the postsynaptic membrane. That's the membrane of that next cell in line. Again, that membrane could be part of a muscle fiber, it could be part of a gland, it could be part of another neuron. A synapse, though, that term is used to describe those 3 major parts. Again, the connection between a neuron and the next cell in line. When we use a more specific term called the neuromuscular junction, which of course is already spelled out in your lecture notes on page 82, that is a specialized synapse. It still has the 3 components, however, it's specialized in the 2 types of cells that are connected. A neuromuscular junction is specialized because it's a specialized connection between a motor neuron and specifically a skeletal muscle fiber. That's when we use the term neuromuscular junction. When you looked at your cell model, your neuromuscular junction model as part of your muscle lab, that's what you're looking at here on the side. This area right here was your synapse. Here in gray and brown, these are your axon or presynaptic terminals. That white squiggly line right there, that is your postsynaptic membrane. It's squiggly because we want to increase the surface area to maximize the amount of space to accommodate our acetylcholine receptors. And then we've got this microscopic crevice right here shown in pink. That pink little crevice right there, that is your synaptic cleft. Those 3 areas together, that's what makes up the neuromuscular junction. So what we need to do next is we need to learn how we can get the motor neuron action potential to the skeletal muscle fiber action potential. And the way we do that is we do that through something called electrochemical signaling. We have an electrical component and we have a chemical component. The electrical component is going to be the story that you learned in part 1. That is the action potential. What we'll do is we'll have an action potential in the motor neuron. We'll convert that electrical signal here into a chemical signal. The chemical signal will allow us to breach or span the synaptic cleft. The chemicals that we're going to be using are those ligands that we learned also in part 1 called neurotransmitters. Then what we'll do is we'll convert that signal back to an electrical signal, and that'll eventually produce our mechanical outcome that we call muscle contraction in part 3. So again, electrochemical signaling. Electrical component is the action potential. Chemical component is the neurotransmitter, back to an electrical signal here in the skeletal muscle fiber. So how does that work? Well, in order to understand how that works, we need to understand and introduce some of our players. Our first player is going to be these guys right here. We call them synaptic vesicles. We've heard that term vesicle before. Vesicle is the same thing. It's just a membrane-bound sac. We use the term synaptic vesicle just to describe the vesicles here at the synapse. What's inside those vesicles are those chemical signals, those chemical ligands that we call neurotransmitters. And there are thousands of neurotransmitters per synaptic vesicle. The specific neurotransmitter that we use, however, at the neuromuscular junction is acetylcholine. Acetylcholine, as we abbreviate ACH, and you can use that abbreviation as well, that is the excitatory neurotransmitter at the neuromuscular junction. What we're going to do is we're going to use acetylcholine, or ACH, again to get us across the synaptic cleft. So as a result, we need to introduce another component here on that postsynaptic membrane. In order for acetylcholine to have any effect on the skeletal muscle fiber, it's got to be able to bind its receptor. So what we have here in black are exactly those, the acetylcholine receptors. The acetylcholine receptors, like any receptor, have specificity. Acetylcholine receptors can only bind acetylcholine. They cannot bind any other neurotransmitter. They cannot bind GABA or serotonin or norepinephrine. They are highly specific for acetylcholine. Another player that we need to introduce are these guys right here. These are the voltage-gated calcium channels. We already should know what that term voltage-gated means. It means it's going to open and close in response to a change in voltage. When it opens, it allows this ion, because again there's specificity called calcium, to move into this presynaptic terminal. If it's closed, the calcium cannot move. What are the voltages that open and close the voltage-gated calcium channels? You may already predict. When the motor neuron is at rest right here, the membrane potential, the resting membrane potential to be more specific, is exactly the same as in our drawing in part 1. It's at negative 70 millivolts. However, when that action potential propagates down the axon and arrives here at the presynaptic terminals, what happens is we change that membrane potential to that plus 30. Therefore, it's the plus 30 that opens up these channels. So negative 70 millivolts, the channels are closed. At a positive 30 millivolts, the channels open. So now that we understand the players, the structural components here to our synapse that we call a neuromuscular junction, let's look at the physiology of the neuromuscular junction. Again, how do we get our signal, the action potential, from the motor neuron to the skeletal muscle fiber? Well, what's going to happen is that all the way up here, again in the cell body of the dendrites, we're going to have other neurotransmitters that bind onto the receptor sites at those locations. Again, the receptor sites are the same receptor sites we learned in part 1. They are ligand-gated sodium channels. Because sodium has a high concentration outside, low concentration inside, sodium will start to leak in, producing local depolarizations. If we get enough neurotransmitter binding to produce enough local depolarizations, what will happen is we'll eventually hit threshold. Once we hit threshold, of course, the voltage-gated sodium channels open, sodium floods the cell, we get that rapid depolarization. What we've done is we've initiated an action potential right here at the top of the axon. That action potential will propagate down the axon until it arrives at the presynaptic terminals. That's what you see right here. When I draw my little lightning bolt right here, the action potential represents a wave of positive charge, that plus 30. Once the action potential arrives at the presynaptic terminal, that is again the signal to open up the voltage-gated calcium channels. As we open up the voltage-gated calcium channels, calcium is now able to diffuse into the presynaptic terminals. Why is calcium important? Because that's what allows the synaptic vesicles to fuse with the membrane to release the neurotransmitter into the synaptic cleft via exocytosis. You may remember back from our study of the skeletal system, we talked about calcium homeostasis. We said calcium homeostasis was involved in a couple important body functions. One was muscle contraction, another was nerve cell communication. If we do not have adequate amounts of calcium, then what will happen is we'll never allow the synaptic vesicles in any of our neurons to fuse with our membrane to release the neurotransmitters. So basically nervous system communication will shut down. Nevertheless, in this case, what happens is, again, that action potential arrived at the presynaptic terminal. It opened up the voltage-gated calcium channels. Calcium diffuses in, and that allows the synaptic vesicles to fuse with their membrane, releasing their neurotransmitter here in red, that is acetylcholine, into the synaptic cleft via exocytosis. Because we have a high concentration here and a low concentration here, the acetylcholine will diffuse across the cleft and bind its receptor on the postsynaptic membrane. Again, here is your chemical component of the electrochemical signaling. As we bind these receptors on the postsynaptic membrane, this is actually the sarcolemma of your skeletal muscle fiber, these receptors again are actually ligand-gated sodium channels. Because we have a high concentration of sodium on the outside and a low concentration on the inside, sodium will start to leak in. As sodium starts to leak in, we're introducing positive charge to inside the skeletal muscle fiber. Membrane potential will move from -70 to -69. We call that a local depolarization. As more and more neurotransmitter binds to more and more of the acetylcholine receptors, we introduce more and more positive charge. More sodium will start to leak in, producing more local depolarizations. We move from -70 to -69 to -68 to -67 and so on and so on. Although I describe that as a single 1-to-1 ratio, that's not anywhere close to being the truth. There are actually millions of acetylcholine receptors here, which is why you saw in the cell model that white squiggly line. This membrane is actually highly convoluted to accommodate all those millions and millions of acetylcholine receptors, but you get the idea. What we're doing is we're slowly progressing in a stepwise fashion from -70 to -65. If we get enough acetylcholine, though, binding enough other receptors, then again, we open up enough of the ligand-gated sodium channels to produce enough local depolarizations, we will hit threshold. At -65 millivolts, the voltage-gated sodium channels here in the membrane will open. Again, because sodium has a high concentration outside, low concentration inside, we get a huge influx of sodium. Because sodium is positively charged, we get that flooding of positive charge, membrane potential skyrockets to +30, and now we've initiated an action potential in the skeletal muscle fiber. So again, we've used electrochemical signaling to get the action potential from the motor neuron to the skeletal muscle fiber. We had our electrical signal in the motor neuron, chemical signal here with our neurotransmitter to get us across the cleft, and now we're back to our electrical signal in the skeletal muscle fiber. The last thing we want to mention before we do a little housekeeping is, do we want this neurotransmitter to be active for basically as long as it wants, for extended periods of time? And the simple answer is no. That's because we lose regulation, we lose control. So therefore, these neurotransmitters, they are active for very short periods of time. For example, for the sake of simplicity, let's say that a neurotransmitter is active for only 1 second. But let's say, though, that we needed 10 seconds of activity. Could we build up to 10 seconds? Yeah, we could. All we have to do is send 10 signals. However, in the contrary, if we had a neurotransmitter that was active for 10 seconds, but the minimum duration of activity was 10 seconds, but we only need 1 second of muscle contraction, could we ever take that 10 down to 1? The answer is no. So again, what we do is we keep these neurotransmitters active for very short periods of time. So as soon as the neurotransmitter binds to its receptor, it unbinds. And then basically we break it down right away so that we can maximize control. Again, if we need continued stimulation, then all we have to do is we have to send more neurotransmitter. We send more signals. The neurotransmitter that is used to break down acetylcholine is called acetylcholine esterase, ACHE. You can use the abbreviation here as well. You don't actually have to spell it out. What acetylcholine esterase does to acetylcholine is basically it splits it in two. It breaks it down into something called acetic acid and choline. You don't need to know anything about that, but basically we change the structure, we change the function, and acetylcholine is now inactive. So with that said, let's do a little housekeeping like we did in part 1. Let's go over our lecture notes. I want to show you a quick animation, and then I want to talk about some clinical applications related directly here to the neuromuscular junction. At the bottom of page 82, we have our neuromuscular junction, also called a synapse. As we said, it's the transfer site of the motor neuron action potential to skeletal muscle cell action potential. Each muscle fiber is stimulated by a terminal branch of an axon. The structures or areas, they include the presynaptic terminal, also called the axon terminal, with the synaptic vesicles. The synaptic vesicles are the membrane-bound sacs that contain the neurotransmitters, thousands of them per vesicle. And again, acetylcholine or ACH is the excitatory neurotransmitter of the neuromuscular junction. We have the synaptic cleft right here, that microscopic space that separates one neuron from the next cell in line. And we have our postsynaptic membrane. When it comes to the neuromuscular junction, we also call that the motor end plate. Top of page 83. Physiology of the neuromuscular junction. The same steps that I just gave to you a second ago. The action potential will propagate down the axon of the motor neuron and arrives at the presynaptic terminal. The voltage change, that's the depolarization to a +30, causes the voltage-gated calcium channels to open and calcium diffuses inward. This causes the synaptic vesicles to fuse with the axon membrane and release their neurotransmitter ACH into the synaptic cleft via exocytosis. ACH then diffuses across the cleft and binds onto its receptor, which is actually a ligand-gated sodium channel on the postsynaptic cell, as you see right here in black. The area on the sarcolemma of the muscle fiber where this occurs is termed the motor end plate, and is highly convoluted to increase the surface area so that it can accommodate the millions of acetylcholine receptors that are located there. The ligand neurotransmitter receptor interaction is very specific, again, as we just said a few minutes ago. ACH can only bind to ACH receptors. It cannot bind receptors for GABA, histamine, serotonin, norepinephrine, or any of the other neurotransmitters. Meanwhile, the ligand-gated sodium channels on the sarcolemma of the muscle fiber open and sodium diffuses inward. This produces a local depolarization of the skeletal muscle fiber, and if enough local depolarizations are achieved to reach threshold, an action potential is initiated in the skeletal muscle fiber. Meanwhile, again, to maximize control, the neurotransmitter effects are then terminated. The binding of the neurotransmitter to its receptor is reversible, and its effects generally last only a few milliseconds before being terminated. ACH is quickly broken down to acetic acid and choline by an enzyme called acetylcholine esterase, and this removal of ACH prevents continued and most likely undesirable muscle fiber contraction in the absence of additional nervous system stimulation. As you can see, you've got your drawings now in front of you. I encourage you to draw those out on your own, and also all the steps already in your notes. This again is part 2 of our story leading to skeletal muscle contraction. To review this, what I'd like to do next is to show you a quick animation. To remind ourselves where these animations are located, again, you can head to my website. You click on More over here, and then Textbook Companion Sites. When you click on Textbook Companion Sites, a new window opens. What we're on over here is Chapter 9, and there's a drop down over here that allows you to scroll down to Chapter 9. As we look here at this animation, we're looking at, again, basically a schematic of the drawing we just made. Here in yellow, this is going to be your motor neuron. This is your axon. That is your axon or presynaptic terminal. In green here, we have our voltage-gated calcium channels. These are our synaptic vesicles with our neurotransmitter called acetylcholine located inside. We have our synaptic cleft. We also have our postsynaptic membrane. These pink or purple guys right here, these are your acetylcholine receptors, which again are actually ligand-gated sodium channels. So let's take a look.
An action potential arrives at the presynaptic terminal, causing voltage-gated calcium ion channels to open, increasing the calcium ion permeability of the presynaptic terminal cell membrane. Calcium ions enter the presynaptic terminal and cause vesicles to release their neurotransmitter acetylcholine from the synaptic vesicles into the presynaptic cleft. Diffusion of acetylcholine across the synaptic cleft and binding of acetylcholine to acetylcholine receptors on the postsynaptic muscle fiber membrane causes an increase in the permeability of ligand-gated sodium ion channels. The movement of sodium ions into the muscle cell results in depolarization of the postsynaptic membrane. Once threshold has been reached, a postsynaptic potential is generated and is propagated over the muscle cell membrane. Acetylcholine is broken down to acetic acid and choline in the synaptic cleft by the enzyme acetylcholine esterase. The choline is reabsorbed by the presynaptic terminal and combined with acetic acid to form more acetylcholine, which enters the synaptic vesicles.
Apologize for the sound quality and the internet connection, but to review that one more time. Again, what happens is that action potential is going to propagate down the axon, arriving here at the presynaptic terminal. That's going to cause the voltage-gated calcium channels to open. Calcium diffuses inward, causing the synaptic vesicles to fuse with their membrane, releasing their neurotransmitter acetylcholine into the synaptic cleft via exocytosis. Acetylcholine will bind onto its receptor site, which is actually a ligand-gated sodium channel. The receptor opens, sodium diffuses inward, causing a local depolarization. If we get enough local depolarizations to reach threshold, then an action potential will be initiated in the skeletal muscle fiber, as you can see right there in yellow, and then we'll break down the acetylcholine by acetylcholine esterase. So folks, between that animation, our drawings, as well as what we have in the lecture notes, we have a pretty good summary of what happens across the neuromuscular junction. What we want to move on to next are some clinical examples before we look at part 3, which will be what occurs inside the skeletal muscle fiber. Bottom of page 83 and on page 84, let's view some of these clinical examples. As we'll see also in the nervous system, guys, what happens here at these synapses, these become primary sites of focus for pharmaceutical companies. Pharmaceutical companies love to design drugs that target these receptors as well as the neurotransmitters. Because this affects nerve cell communication, by affecting or impacting the receptors and the neurotransmitters, and the nervous system being involved in control of all of our body functions, we can use these pharmaceuticals to actually have an impact on body functions all throughout the human body. We're going to focus strictly here on the neuromuscular junction for now. But again, many toxins, drugs, and diseases do interfere not only at the synapse, but at the neuromuscular junction. Based upon what we've already learned, we probably can actually use some common sense to predict what these or the impact of these would be. So let's take a look at some of these examples. If we were to limit acetylcholine esterase activity, what do you think would happen? If acetylcholine esterase, though, was not able to function, would we have any impact on acetylcholine? And the answer would be no. Acetylcholine, therefore, would be able to stay active. Because it's able to stay active, there's no way to turn it off. This results in continued stimulation of the muscle fiber, something that we call spastic paralysis. With spastic paralysis, though, we are able to contract, but we are not able to relax. This will eventually end up with muscular fatigue. That's where the muscle stops working and is not able to contract any longer. The end consequence, though, is death through respiratory failure. That dome-shaped muscle we learned in our muscle lab called the diaphragm has to be able to contract as well as relax in order to function. So therefore, if it cannot contract or relax, in this case, though, that is what happens if we limit acetylcholine esterase. Again, we're unable to breathe. We end up with death through respiratory failure. Organophosphate pesticides and some nerve gases, they target acetylcholine esterase. That's why you see a lot of our farm and field workers wearing the respiratory devices. Other agents may target or even block or damage acetylcholine receptors. If we block or damage an acetylcholine receptor, what happens is we filled up the parking spots. If these receptor sites are already filled, then acetylcholine cannot bind, so therefore we have no ability to stimulate contraction. This results in the opposite of spastic paralysis. We call this flaccid paralysis. Flaccid paralysis is basically the patient, though, is laying on the floor in front of you. They cannot speak, they cannot move, they cannot breathe. Again, death oftentimes to respiratory failure. This is how, though, both snake venom and anesthetics work. Anesthetics, of course, in a controlled environment, what we do during a surgery, of course, we don't want you to move, so we temporarily paralyze you. We use agents, however, though, that can bind and unbind onto these acetylcholine receptors. What we do with those agents is we fill all the parking spots, that is to say, we fill the receptor sites. Acetylcholine cannot bind, so you cannot move during a surgical procedure. However, because this is in a controlled setting and that those agents can bind reversibly, which means they can bind onto the receptor site and then unbind, once we stop administering the anesthesia, then those molecules come off the receptor sites, acetylcholine can go ahead and rebind, and now we are able to resustain muscle contraction. Of course, we're going to make sure our patient is able to contract their muscles on their own before we ever let them go home and leave. But again, that's how we take care of with anesthetics. Snake venom is a little bit of a different beast. It also binds the acetylcholine receptors, but unlike anesthetics and some of these other agents that are able to bind and unbind, and if, by the way, if they're able to bind and unbind, they're using weak chemical bonds, not strong ones. Snake venom, what it does, it binds on with covalent bonds. So when snake venom gets in the blood and starts to find these acetylcholine receptor sites, it latches on and it does not let go. That's why we need to get the antivenom into the bloodstream as soon as possible so we can mop it up before it ever has a chance to bind these acetylcholine receptors. If we don't catch that in time, again, death through respiratory failure. Another disease that affects the neuromuscular junction is called myasthenia gravis. This is characterized by such things such as drooping eyelids, difficulty in swallowing and talking, and overall generalized muscle weakness. What happens here is that it's believed to be an autoimmune disease where our body starts to target our own acetylcholine receptors. Because of the shortage of acetylcholine receptors, we're not going to get proper muscle contraction. And if we don't use our muscles, we lose them, so we end up with much muscular atrophy, and that's why you see some of the symptoms that I just described a second ago. What happens is that, interestingly enough, in order to treat the disease, what we want to do is we want to preserve as many acetylcholine receptors as possible. So sometimes we'll actually use acetylcholine esterase as a medication. What it does, it keeps more acetylcholine into the cleft for longer periods of time. We get more binding on the receptors, which prevents and slows down the attack by the immune system on those receptors, and it ensures, though, more contraction of our skeletal muscle fibers, keeping them healthier for a longer period of time. Tetanus toxin. Tetanus toxin actually doesn't target the neuromuscular junction itself. It actually targets regulatory neurons way up in the brain spinal cord that control the release of CH from the presynaptic motor neurons. Essentially, what we're doing is we're taking our foot off the brake. Because of that, we are flooding the cleft with acetylcholine. Because we flood the cleft with acetylcholine, what we do is we end up with continued stimulation of the skeletal muscle fiber, again, what we call spastic paralysis. What happens is the muscles contract, they're not able to relax until we run out of energy, and that eventually results in muscular fatigue. Again, death through respiratory failure because we're not able to both contract and relax the diaphragm. Finally, botulism toxin. Botulism toxin actually blocks the release of acetylcholine from the presynaptic cell. The overall effect is the same as we talked about just a second ago if we were to block the receptors. With no acetylcholine, we have nothing to bind the receptors, so we have no way to achieve threshold in that skeletal muscle fiber. Because of that, we have no way to stimulate muscle contraction. So again, flaccid paralysis, no muscle fiber stimulation, death through respiratory failure. All right, everyone. Well, that concludes part 2 here of this 3-part sequence on our Muscle 2 lecture. Don't forget also, you've got my drawings, you've got the animation, you've got what's in the lecture notes. You also have your textbook. They've got some good figures as well. What they've done for us here is they've blown up the neuromuscular junction. Look at here in yellow at your presynaptic or axon terminal. You've got your synaptic cleft. You've got your postsynaptic membrane with the acetylcholine receptors. Our voltage-gated calcium channels are synaptic vesicles with acetylcholine. So again, don't be afraid to use your textbook as well. What you want to do, though, before you move on to part 3 is make sure that you have both parts 1 and part 2 of this 3-segment sequence familiar and memorized. What we're going to do in part 3 then is we're going to build off this story here in part 2, and then we're going to see how that actual potential that we just generated down here in the skeletal muscle fiber will produce a mechanical outcome. Enjoy your studying. Again, if you need anything or have any questions, please don't hesitate to reach out. Otherwise, I'll join you shortly for part 3.